Hi, I'm Amy Soto. This is
Valeria Maldonado.
and we are presenting our problem-based project. We are students in Professor Juan Morata's microbiology lab. I mean, there we go. So we are presenting a project based on identifying a bacteria. We used microbiological techniques, and this is important because it helped us understand microbiological diversity, also like figuring out how to diagnose infections and targeting certain treatments as we learned further throughout the class. Through gram staining, we were able to figure out that our unknown bacteria was a gram negative at first. After doing further tests such as lactose fermentation and oxidase testing, we both got negative results in that. And then after we did an indole test and we got a positive result. So after doing all those tests, we were able to identify that our bacteria was a Proteus vulgaris. And yeah,
So the purpose of this study is to identify unknown bacteria using biochemical testing. We used AI and microbiology lab tools during the biochemical test provided initial understanding of the bacteria. We used AI to create a dichotomous key. The standard lab tools ensured proper filtering and isolation of the bacteria. It allowed us to observe and validations.
this is our dichotomous key. This is just a broken down version of just a map of how we figured out our unknown. So after, like I said, and how Valeria said, we used AI to help us create our dichotomous key, and that helped us form just to like break it down and make it simple for us. And then we did actual testing to confirm our results. So as you can see, I had unknown bacteria number 7 after doing gram staining. We got a gram negative. After that, we got a negative lactose result. After the lactose test, we got a negative oxidase test. And then our last test was a positive indole test, which resulted in Proteus vulgaris.
Some materials that we used were agar plates, Bunsen burners, microscope slides, enucleating loops, Bunsen burners, crystal violet stain, alcohol, saffron, distilled water, staining tray, and bubbleless paper.
So some of the methods we used was gram staining. In our gram staining technique, we used that just pretty much differentiates different bacteria we have. So you have a gram positive or a gram negative. If it comes out purple, it is a gram positive. If it's negative, it's a pinkish color based on these cell wall properties. The process of gram staining involves using crystal violet with iodine, and we decolorize with alcohol, and then we counter stain with saccharin. After our gram staining, like I said, we use a lactose test, and through the lactose test, we determine the bacterium's ability to ferment lactose. So that is producing an acid or gas. Bacteria are cultured in a medium containing lactose and a pH indicator. A color change indicates acid production. And then after the lactose test, we did an oxidase test, and that identifies the bacteria by a color change. So after we add the reagent, if it turns a dark blue or purple within seconds, it is a positive result. And then if it does not, then it is a negative result. And then after adding the reagent, we do a indole test, and that detects the ability to determine the bacteria's. But after adding the reagent, if you have a positive or negative result, we had a negative result, meaning we had no color. After that, we did an indole test, and the indole test it detects if the bacteria has the ability to produce indole. After the bacteria is cultured, we add a Kovacs reagent, and then if it has a red layer on top, it indicates a positive result, which is what we had. To conclude, after a series of biochemical tests and different results, we were able to get our unknown, which was Proteus vulgaris, as we've mentioned before. Those are our test results: gram negative, lactose fermentation, oxidase test, and indole, which all match the characteristics of P. vulgaris. The emphasis of this project is to have careful observation and methodical testing of the microorganisms and being able to identify them. Combining all these tests essentially helps us distinguish the differences between the bacteria and if they have similar traits. And what we learned is that we learned how to enhance our microbial classification understanding and biochemical testing in microbiology. These are our references.
We got shared a folder on Google Drive by Professor Juan Morata. We also used the microbiology book given by school, and we use Copilot as our AI source. Further research or acknowledgement. For further future biochemical tests, additional tests can be conducted to further confirm the identity of the bacterial sample. This research is funded by Cresting Opportunities for Applied Learning Experience in Science, Computing, and Engineering, a program of STEM Legacy Grant Project at Miami-Dade College Wilson campus.
For your time and thank you for this opportunity. We wish you well. Thank you.
Bye.